Thank you so much for the wonderful introduction. I am pleased and really honored to be here. We're going to talk about returning to operation following the SARS pandemic. So this is my disclosure. So just to get started, so really, as we all know, this is just a few very obvious things for all of us, in that we exist, in the current pandemic that has gripped all of society in so many ways. And so IVF laboratories at this point in time have all been shut down at varying degrees, whether they shut down immediately or were able to finish their IVF cases that they did. The point is, many labs have been closed down much longer than they thought. So I thought that I would take this opportunity to discuss these four major topics: cleaning the exterior of the laboratory, and I'm going to diverge a little bit to talk about some viral transmission that's potential mostly in large animals, because I think that's a good model. I want to talk about our HVAC systems that exist today, because I feel like HVAC systems could potentially be an area that we could have ongoing contamination. And then we're going to talk about how do we clean the inside of the lab, what are the areas of concern, and then QC process to ensure that the lab is fully functional, and then how do we reopen the labs today in our centers today, and doing it as a good stewards and good citizens. So really, my goal today is really to provide some suggestions that could be used really anytime. So in other words, I'm hoping that I could provide some suggestions for people that maybe have not thought of them. So we're going to get pretty organic in certain areas. If this is something that you already know about or know a whole lot of, I apologize. But really, the whole goal of this is to try to take this time and try to reinvent the processes that we're doing and reinvent what we do. And then including with that, we're going to talk about quality control, how to improve our quality control processes. So as we all know, this beautiful little virus has been creating a lot of havoc for us. It's a single-strand RNA virus. I'm not really going to get into it, what it is, because we've had some amazing lectures earlier about that. But what I want to stress is that there's limited ability of this virus to bind because it requires an ACE2 receptor, but this receptor site has been found, as listed in one recent paper, in some of the testicular tissue, as well as found in developing spermatogonia. But up to this point, we've really had no viral transmission or no vertical transmission yet. So I want to talk a little bit about the zona pellucida, because this is a structure that all of us in the laboratory at least know what it looks like. But really, I thought might be a good opportunity to revisit what is the zona. Well, the zona is really this complex structure that's made up of 3 sulfated glycoproteins. The zona really acts as a wonderful barrier due to its acellular nature, its thickness, and the lack of receptor sites that are present on there. What's interesting, though, once you look at the zona from an electron microscopy perspective, you see that it's very spongy-like, that it's a complex network that has many, many pores, and these are the pores that existed are remnants of how the granulosa cell is able to communicate with the oocyte prior to ovulation. So what happens is that these pores decrease in size as you get closer towards the interior aspect of the oocyte, and these pores change in the zona dramatically following ovulation and fertilization. So the information that we know about pathogens crossing the zona pellucida is really very, very low. So this is an electron microscopy imaging of 3 zona pellucidas. We have the bovine zona pellucida, and what's interesting here in the porcine and the human, what's interesting is that the size in the holes of the zona, this is the exterior aspect of the zona, range 2 to 300, 400 nanometers in the human to almost 1,000 nanometers or 1 micron in a porcine oocyte, whereas in the human, these pore sizes tend to be all under 192 or under 200 nanometers. If we look at the viruses, though, below, you'll see that a bovine BVD virus, this virus can be as small as 60 nanometers all the way up to the size of the human coronavirus that we're dealing with is the size of 120 nanometers. So we see that the virus that we're dealing with right now is a pretty large virus. Now this is really good because large viruses are much easier to remove through centrifugation than smaller viruses are. But I just wanted to show you that what the zona pellucida looks like. It's just not, a bunch of holes in a zona. It's a fairly complex architect in comparison to the viruses that are commonly found in these host animals. So why are animals a good model for all this? Well, really, the reality of it is there's very little data that exists in human studies. But we can all take great comfort in knowing that before 2005, there were many, many embryos that were transferred in donor oocyte models, and we had no known diseases transferred. There are some literature sightings of HIV found in poorly prepared semen samples, but that's really what we know right now with humans. So really, if we want to now take some time and learn about this, studying animals is really probably one of the best models to study as far as how it relates to humans. And the reason for that is that we can take these animal embryos, expose them to pathogens at different levels, and we can transfer these embryos to recipients, or we can take animals that are serologically positive for certain viruses, mate them, transfer those embryos to serologically negative recipients, and see what the outcomes are of that. And in one of the first papers, it was Singh et al., 1982, this author worked at the time at the Agricultural Research Center up in Canada, which has done a marvelous job along with Stringfellow and other authors to really study the effects of viral and bacterial transmission, the potential of that through embryo transfer. Well, in this particular study, which is kind of considered a landmark, they were taking embryos at various stages from 16-cell to the blastocyst stage. We're exposing them to various levels of this virus, the IBRV, and there was no evidence that the virus could be transmitted through the zona. And what was found, interestingly, and this is one of the first papers talking about the concept of washing the embryo, and that washing the embryo effectively removed this virus from the zona pellucida. Embryos that were collected from serologically positive donors and that were transferred to recipients also were negative. So this study showed that the virus could not infect the embryos and could also be removed and was not going to infect the recipients. And this kind of theme exists with the next one. So I thought, okay, well, viruses are big. What's the next smallest thing we can look at? So now we looked at the prion that causes BSE. And this paper from Rathall et al. And veterinary records show that basically they were not able to transmit BSE to recipient animals. These recipient animals were allowed to grow for a long time. The babies that were born also did not exist. That they did necropsy of the brains and never did find any evidence of this virus at all in any of the spinal fluid or brains of any of these animals. And then last but not least, I chose the goats because the goat the embryos are collected surgically. The ability for there to be a lot more fluid transmitted and stuff like that is kind of maybe more or less looks more like what we do in an IVF lab, although not exactly. But the interesting thing is these embryos were collected, they were frozen, and then transferred to CAEV-free recipients, and the offsprings as well as the recipients were negative to this virus. So, you can look at a lot of papers and find that the vast majority of the literature states that there's really not a lot of issues with this. So the studies really seem to indicate that the virus doesn't readily transmit, and that's probably due to the lack of receptor sites as well as to the actual physiological structure of the zona, as well as the changes that occur to the zona pellucida following ovulation and fertilization. And then we know, because in the embryo transfer industry with cattle, that viruses can easily be removed from the zona pellucida, and in fact, this is something that's required of all embryos prior to them being frozen. Now, one thing that has come up in reading these papers are that if there was a theoretical method of transferring a virus to an animal was by transferring hatching blastocysts or zona pellucidas that are compromised. So maybe we need to maybe we should go back and revisit not using ICSI so much, and perhaps we could go back to doing a little bit more conventional inseminations. Again, this is an example of where we could maybe rethink things. So these are my own personal recommendations, but I have to tell you that it's out there. We have a paper that was published in Cryobiology that theoretically a liquid nitrogen tank can be the source of contaminants. We have found spores in virus, or I should say in the vapor phase. That until we understand this virus better, I would suggest trying to freeze oocytes, embryos in closed devices. If need be, we found out that this virus was indeed present, we could always develop enzymatic cleaning like trypsin, which is used a lot of times with cattle embryos. And then maybe for the time being, to be super conservative, that maybe these gametes should be quarantined until we decide as a society what's best. So now we're going to turn our attention a little bit to HVAC systems. And I'm going to break HVAC systems down into two basic types: closed systems and open systems. Closed HVAC systems are the ones that we see a lot more now. They're characterized by really high air exchanges, elaborate air filtration systems, and positive pressure relative to the rest of the clinic. Whereas open systems are found more in older settings, a lot of times in office IVF lab settings. What these all have in common, though, is that the ductwork is shared, you have limited air exchanges, and you have minimum air filtration, and usually pressure is neutral relative to the other rooms. So if we compare and contrast, closed HVAC systems have very robust ductwork because they have to handle the increased pressure. They also have advanced filtration systems, so carbon and potassium permanganate is a hostile medium, I should say, for viruses, so it tends to kill the viruses. And the air exchanges are usually 16 to 18 hours air exchanges per hour, so we're going to very effectively disrupt any theoretical aerosol that's present in a laboratory, for example. And the positive pressure that you have in these rooms is going to reduce the potential for contaminated air to enter. So really what it's saying is that the theoretical viral introduction is greatly reduced in these kind of places, in these kind of scenarios, because labs that are built with these type of HVAC systems are also built for self-isolation so they can have positive pressure. So what about open HVAC systems? Here they tend to have shared ductwork with the majority of the building, so there's many sources of contamination. Since the ductwork is not sealed because it doesn't have to handle high pressure per square inch, these ductwork can be a source of leaking. We have minimal air filtration also, so they tend not to kill the viruses. And the air exchanges an hour are typically less than 12, so that's really not sufficient to remove the aerosol that the lab staff are going to produce. And the other thing about it is these rooms tend to lack positive pressure, so the potential for outside air to enter the lab every time you open and close the door is great. So what can we do as far as just how to compare and contrast our labs as far as the HVACs go? Well, on the left are the closed system and on the right is the open system. So a lot of these things we discuss, but really these labs are best because they're going to literally scrub the air. They reduce outside contamination. They really provide the proper air exchanges that laboratory staff really need. And they provide positive pressure, which also decreases the amount of contaminants that can come in from the outside. So what can we do to improve this kind of HVAC system? Well, we can be cognizant of changing the filters, because exhausted filters are not going to filter the air and therefore not going to remove viruses. And the other thing that we can do is change our scrubs every day, because a lot of people seem to wear scrubs multiple times. They wear them home, they wear them back to work, and now you're introducing a source of contamination in a closed system. What about an open system, though? There's really a lot of things that we can do here, though, to help out. Before you inspect it, though, I suggest that you shut the system off and that you wear, PPE and gloves when you're doing this, because there's a lot of sources of contamination. So some of the things that you should look at, try to improving is upgrading your fan motors if you can. And realize if you do have HEPA filtration in these scenarios, they don't help. You need to install nanofiltration. In other words, where you're filtering down to the nanofilter. If you do that, though, it's going to require quite an upgrade in fan motor to deal with the added resistance that you're going to get. If possible, do that. If you can't, then try to do things like increase your positive pressure. That's going to often mean doing things like balancing the air in the laboratory. Increase your air exchanges an hour so that you remove aerosol as quick as you can. Replace these filters as often as you can. And make sure you clean the diffusers with something like a 70% ethanol or a 6 to 10% hydrogen peroxide. The other things that you can try to do is reduce staffing in the lab, because, the air exchanges are going to be if you don't do that, then this could be a potential source of contamination. And like I said earlier, change your scrubs. So now we're going to talk about cleaning the laboratory, and the approach that we're going to take here is we're going to start on the outside and work our way into the laboratory. And my thoughts on this are really, you want to use multiple cleaners, and this is going to have, and the reason why multiple cleaners are that you're going to wipe the area more than once, so you're much more likely to get, any potential contaminants, whether they be viruses, molds, or yeast. But it also depends on what the surface material is and the sensitivity of the surface materials. Not everything does very well, for example, with hydrogen peroxide. So obviously, where do we start? Well, we should start with our doors and work our way inward. In here, I've kind of made some suggestions of what you can use. So we use these Sani cloth wipes that are a germicidal wipe that contains alcohols and ammonium chlorides, because when these break down, they leave the lab relatively quick. We don't have any embryos in the lab, so we're not really worried about these latching on to any oil that we have in the lab, oil culture dishes. You also really need to clean the floor as well. And again, you can use something that's a combination of alcohols and ammonium chlorides. But again, it's really important that you allow these materials to sit for at least 1 to 2 minutes, because it's a contact that kills the virus or the yeast or the bacteria. Hydrogen peroxide works great, but if you're going to do that, make sure you check the floor first, and by all means wear goggles and a mask. Another area that we often don't clean as well as we should are the collection rooms. And using a 1% 7X solution, this is a detergent that's used very common in tissue culture labs and used in a lot of embryology labs because it's relatively toxic, less toxic to cells. And you want to follow by water on clean materials. So, couches, things like that, clean very well with 7X, but they don't do so well with ethanol or hydrogen peroxide because it tends to discolor them. So those tend to work better on resistant surfaces. But again, the theme of it is you got to let the material sit there at least 1 to 2 minutes. Preoperative rooms, recovery rooms, these are all areas that don't tend to get cleaned as much as they do, because we think they're outside of the critical care area. Well, now during this downtime is a really wonderful time to clean these areas as well. And you can use hydrogen peroxide again. One thing I do want to state here, though, is regardless of what cleaning agent you decide to use, I would suggest that you use a spray product whenever you can. And the reason for that is that the surface stays wet longer. So if you do use products like wipes, you're going to have to use them multiple times because you're going to have to you need to calculate for a 1 to 2 minute surface contact in order to, effectively disinfect your lab. So that leads into the next thing. When you read tissue culture papers, they make a very clear distinction between cleaning a lab and disinfecting a lab. I'm going to kind of use those terms interchangeably, but I am going to tell you this: you cannot disinfect a dirty lab. So clean the lab first before you try to disinfect it, because all you're going to do is disinfect dirty stuff. Like I said earlier, change the HVAC system off if it's a open system. Think about the gloves that you're wearing to clean with. A lot of the cleaners that you may want to use may not necessarily work with the gloves. You don't want to do anything to your hand. These are things that people don't think about until their hands are burning. Clean from the top down. Light fixtures are really effectively cleaned with ethanol. How do you clean your walls? This is a great question. I'm not really sure how to answer that question because a lot of that's going to depend on is it an oil-based or a water-based? If it's water-based, you really can't use anything on that or else you're going to discolor the paint. If it's an oil-based, which I hope you don't have, but on that surface you can use a lot of different cleaners. The floors should be cleaned again, just like I stated in earlier. Again here, now that you're actually inside your facility cleaning, you really need to think about what to clean with. Don't use glutaraldehydes. And the reason for that is that glutaraldehydes are really effective cleaners. In fact, glutaraldehydes are used a lot for coal sterilization. The problem with glutaraldehydes is that they break down and they form these radicals that are extremely embryotoxic, and they persist for a long time in the air. And in fact, in tissue culture labs, most labs don't use them because the glutaraldehydes end up impacting cell culture development after a cleaning. So please don't use glutaraldehydes. After you turn your HVAC on, allow it 1 to 2 days to completely clean out and get new air in there. If it's an open system, if you have a closed system, overnight is more than enough time. So now that you've cleaned the lab and now you're standing there, the next thing I would do is kind of look around at your equipment and go, okay, what's the highest potential? What's my lowest potential? And water baths, box incubators, circulating water baths, incubator fans, all of these things are areas that have high potential, and these should be cleaned as I've listed here in the slide, because these are great sources of bacterial and fungal contamination. So, this is time to do that. Hoods and surfaces tend to be lower potential for contamination, so we tend to clean those with 70% ethanol, followed by hydrogen peroxide, and again, we're letting it sit. And something that's very common in tissue culture labs, but not so much in embryology labs, is use overlapping motions. When you clean with one solution over another, work at 90-degree angles so that you always have overlap. Change your hood filters. Benchtop incubators are really easy to clean. If depending on what type of water reserve they have, you either replace it or clean it, sterilize it. If you have a plastic water reserve, make sure you fill it with water, let them off gas, because many times you will fail a mouse assay if you don't rinse and off gas these bottles. Centrifuges are an area of great high potential, and the reason for that is centrifuges don't tend to get cleaned as much as they should be. If you do clean your centrifuges, though, make sure you don't use alcohol on the rotors. That's a big no-no. And clean the inside of them very, very well. I clean them at least 2 times with hydrogen peroxide because these are a great source of contamination. What about other things in the lab that we touch all the time? These are things like injectors, control pads, computers, scopes. Depending on what the material is, either clean it again with hydrogen peroxide or ethanol, and we tend to do it twice, just to make sure that we overlap. Don't forget to clean the eyepieces on your microscope and knobs. This is something that people don't do. If you were to look at eyepieces are a huge source of contamination between staff members. So eyepieces should really be cleaned at the end of every day. Other things like buttons, flasks, phones in the lab, diamond pins, anything that you touch all the time, probably wouldn't hurt it to give it a good cleaning now as well. So my final thoughts on cleaning are: start with the exterior, work your way down, start at the top. If you must clean, you must clean before you disinfect. Use multiple cleaners, that's what works best. Sprays are more efficient. Concentrate on the equipment that has high humidity levels or water could potentially be standing. Make sure you allow your lab plenty of time to off gas, and that's going to be dependent upon your HVAC system. And also know that your HVAC system moving forward could be a source of contamination. So, as I stated earlier, I wanted to take this time to not only think about how are we going to open again, but what can we do to reinvent ourselves? So reinvent our processes that we use in the lab, and one of them is quality control. So an area that I think is very important is that let's start with our surface temperatures and our culture drop temperatures. Who knows when's the last time you actually did that? I think what I would want to offer to you here is to go through your mock procedures. Make sure that the culture media does not raise too high and err on the cool side. Now's a great time to do QC on your gases and also develop a tracking system for your consumables. And then when you're done with everything and you feel like the lab is ready to open, this is a wonderful opportunity to develop an increased sensitivity mouse assay, so you can make sure that your lab is ready to open. So here's just a few things that we do in our lab. The slide here on the right will show the template of, for example, of a 6-foot hood. So when we go to start QC, we check all the places in the hood where people would potentially put dishes, and then we actually go through mock procedures. And I've given some suggestions here at 0, 5, and 10 minutes. You want to place the dish under the bubbler because you want to actually recreate the events that you're doing. And the reason why this is really important to do is because a lot of us use dishes with different architect on the bottom of the dishes. So some of them have flat bottom and others have raised bottom dishes. And so that's actually going to impact temperature much more than you think. So the take-home message here is really mimic the procedures that you do in the lab. And that's the same thing that we do with our microscopes. Again, I don't know when's the last time you calibrated your heated stage, but we all know that many of them have notorious poor thermodynamics. They heat cycle and can make it worse if they're open and closed. So again, you want to check for hot and cold areas. And again, when we're doing these things in our laboratory, we tend to mimic processes that we're going through in order to see what our temperatures are. So on the right here, our hood or our heated stage has the outer areas heated as well as the inside. So we check all the outer areas for hot and cold spots. And then the interior, we also check for hot and cold spots because we want to make sure that when we're doing ICSI or things like that, that we're not actually getting too lethal with our temperatures. So you would be surprised what oil over the top does to your temperatures. So again, now let's assume your lab has been closed for a while. So if it has been, this is a really good time to flush your gas lines and flush your filters out if your incubators have been off. You may even consider changing the filters that are in your laboratory. And the reason for that is that these filters have accumulated VOCs. So when you turn the gas on, depending on what the VOC load is on these filters, you could actually release a whole lot of VOCs into your lab. The other thing you might want to consider is upgrading the quality of your CO2 and nitrogen, mostly because CO2 is the most contaminated gas in your triple gas mix, and nitrogen because it's the largest percentage of gas in your gas mix. If you have incubators that can mix their own gases and you have your own manifold system, consider using the liquid nitrogen vapor. It's extremely clean, much cleaner than you would ever find in a cylinder. Perform pH testing once your incubators are fully functional, and incorporate pH testing with every new lot of media and gases you get. Consumables, we talk about this a lot, but a lot of people don't have a good inventory system. But buy MEA products whenever you can. Develop sperm assays whenever possible. And our sperm assays are long exposure. In other words, we expose the material in question for up to 72 hours before we actually do a sperm survival test. Off-gas your products. Track when everything comes into place. And if you can, hold lot numbers, because this will really help in your cost, and it'll reduce the introduction of variables into your lab. Some final steps before actually getting your lab ready to open is make sure you've off-gassed enough, test your air if you can, make sure your incubators are up to date, and then go ahead and do an MEA if you can. In our lab, we would use a less complex media like, say, HTF. We use one-cell embryos, and we would turn the oxygen off in our big box incubator to increase stress. We would set up dishes two ways, with oil and without oil, and we would open the incubator daily, mimicking procedures. And this, in our opinion, is going to give us the best indicator of a lab air environment. For your benchtop incubators, you can, again, do the same thing without oil, some with oil, open the incubators, and your endpoints should be, though, well-developed, expanded and hatching blastocyst robust embryos. If you can, try to do some cell counts. But I beg you, please don't start up until the lab passes your standards. So how to reopen the lab now? We're going to touch just a couple slides on this. You really need to develop a social distancing program. You want to rearrange your waiting room. And you really want to think about using the parking lot as your initial waiting room. You want to check temperatures on patients before they come in. And our policy is you must wear PPE, no exceptions. And something that's really important is going to dictate based on your individual needs are what's the amount of staff that you can have. And this is going to be impacted by your air exchanges, because we know that the base reproduction rate, in other words, how many people get infected when we don't practice social distancing, with the SARS COVID-2, it's about 3 to 5 people. Implement cleaning strategies for your exam rooms, common areas, and develop a schedule that's going to reduce or minimize patient-staff contact. Once you've kind of got that in place, how do you open up the clinic itself? I would suggest staggering your IVF cases. Start slow, work your way up. Frozen embryo transfers are a really good way to start. All staff have to wear PPEs. Make sure that you use the waiting room and even the parking lot to stagger areas of congestion like the ultrasound rooms and your blood draws, and clean between cases. And ideally, your shift change should be clinic-wide, although that can be really hard to do. Once the staff and everybody's ready to go, all staff should really wear clean scrubs daily, incorporate sticky mats, shoe covers, masks, open up centrifuge samples under the hood just to deal with the aerosol. And really, I want to implore to you that you can continue to do your embryo procedures as much as you want, because if you think about it, the way we wash gametes, the transfer rate is essentially nothing. And then establish your guidelines for transfer, whether you freeze all or you're going to do transfers. So in conclusion, we really live in an unprecedented time. We don't have all the answers to this. In fact, a lot of these processes we're developing as we go. Again, I want to get back to you though. Cleaning has long-term consequences. Avoid glutaraldehydes. I don't believe that viruses are a big issue for our gametes because the way we wash sperm samples today in gametes, the dilution factor I think is such that it's not going to have an impact. I do think we should be cautious with sperm samples. Should we quarantine sperm oocyte embryos? I don't know, maybe so. But I do want to tell you this, as an embryologist, don't open your lab until it passes at least one bioassay, because remember, the buck stops with you as an embryologist. So, because when the dust settles, you're going to be the person who's going to have to answer to the medical director and all the patients that your lab is ready to go. So I've got some suggested readings here. The AAB had some really great guidelines, and then all these other guidelines about viral hepatitis, assisted reproduction. And then I've included quite a few guidelines that are used commonly in the tissue culture world. So you can find those here. And then last but not least, I want to thank Tim Schimmel, Antonia Gilligan, and Corey Kelly for all the help that they did in helping me get this presentation together. I have to acknowledge the wonderful laboratory, medical, and most amazing nursing staff that we have here at Poma Fertility. And thank you so much for all your time and effort. Thanks for being here, everybody.